So welcome back to the seminar series by the International Tree Mortality Network. We received quite positive feedback from last year's workshops on the future of our network, which at the end now motivated us to continue this seminar series that was on hold since last summer around. In future, as you might see already, the seminar series will be supported by new colleagues. So today, Gina Marano from the WVSL in Switzerland, Martínez de Castillo from the University of Mainz in Germany, and Johanna van Passel from the University of Ghent in Belgium, Robbie Hember from the British Columbia Ministry of Forests in Canada, and Tamir Klein from the Weizmann Institute of Science in Israel are joining. So thanks already now for all your support in the future. When preparing the workshop series last year, we realized that thanks to our speakers, so today it's the 29th seminar, and of course you, the community, we are in fact YouTube stars with more than 10,000 views in total so far. So, and because we want to see that number increasing, we are really happy that Jordi agreed to relaunch the seminar series today. So Jordi, no pressure. I'm sure that most of you from an ecophysiological or forest ecological background are well familiar with the work by Jordi. So I won't go into detail here. And Jordi, in fact, one of the first papers I remember reading was your study on root and stem hydraulics from 2002. So it's quite shocking that that's already quite a while ago. Anyway, before we start, some basic information. Commonly, please use the chat for asking questions. Today we have a normal Zoom meeting, so you're also welcome to raise your hand at the end if you want to ask questions yourself. But normally we will compile the questions, add it to the chat, and then forward them to Jordi at the end. And with that, I will hand over to Jordi. Jordi, the floor is yours.
Okay, welcome everyone, and thanks very much, Bernard, for reminding me how old I am. And yeah, welcome everyone. Thanks to the organizers for inviting me. It's really a pleasure and an honor to be here today. So I will be talking about, I hope you all see my screen. So I will be talking about the limits and opportunities in predicting drought-induced forest mortality, which is of course a topic quite at the center of the International Tree Mortality Network. So basically, I will be dealing with the prediction of where and when these type of events that you are seeing here in the image are occurring. This is from California in 2016. And the question is, are we really able to predict when these type of events occur? So I will start by talking a little bit about why we should care about tree mortality. And with this, I will probably be repeating some aspect or some bits of knowledge that most of the audience knows, but I think it's important to keep those in mind anyway. Then I will go through why it is that tree mortality is so difficult to predict, in particular drought-induced tree mortality. I will then discuss a little bit different approaches to predict mortality that have been used, mostly contrasting trait-based with more empirical approaches. And then I will go into discussing a little bit whether we can combine these two approaches to get the best of the two worlds, and what are the new opportunities that I see arising in that regard. And of course, that will lead me also to some challenges to really realize this potential that we have ahead. And I will end with a very short final note on the importance of global and regional mortality networks, basically presenting the Spanish REDEC, which is a regional tree mortality network similar to the ITMN, but at the Spanish scale. I believe it's quite important that all these initiatives are connected at the end somehow. Okay, so we all know that drought-induced forest mortality is widespread, and the number of events that have been recorded are increasing. And this basically affects forests worldwide. So it's not only something that happens in dry areas, it happens also in the tropics, in the Amazon. So it's really very widespread and potentially quite concerning. It is concerning because for many reasons, but one of them is that we know that forests have a very important role in the carbon cycle. We know that forests are a very important sink of CO2. They absorb roughly around 30% of the CO2 we emit to the atmosphere. We also know from this paper you have here in the screen that this sink capacity of forests globally more or less has stayed constant for the last decades. But we also know that there are risks to the maintenance of this sink capacity, which have to do actually, among many other things, about the disturbances that are themselves related to climate change and how climate change impacts forests. And this is because we know that many of those disturbances are on the rise and will be increasing even more under climate change. And this is the case also for droughts. So you have here data from this recent study of multi-year droughts showing that the area covered by this type of mega droughts have been increasing over the last decades. And this is happening pretty much everywhere on Earth. And of course, we know that these droughts, besides the demographic impacts or the more immediate impacts they also impact the carbon cycle of forests and actually all the biogeochemical cycles in which forests are involved. And we do know that when these events happen, like you have here for the 2005 event in the Amazon, in the figure to the in the left, forests can actually become a source instead of a sink of carbon. And this is even true for forests that we normally don't consider water limited, like rainforests in the Amazon. What is even more concerning is that this 2005 type drought was initially believed to be something that happened around 1 year every 100 years. And actually turns out that we've had similar events of similar magnitude or even higher magnitude in the Amazon, as you can see in the map in the right, in some areas up to 5 times since that initial event in 2005. So there is the clear real risk that this sort of mega droughts will have a substantial impact in the capacity of forests to absorb carbon. So as I said, for these and many other reasons, really being able to predict this vulnerability, which forests are more vulnerable, where, this is becoming a critical research question. And this is important because we also know that forests have are quite resilient. So it's not at all clear a priori which forests are going to be more affected or less, or when these mechanisms of resilience will be broken and will lead to changes in vegetation. The good news is that actually we understand quite a bit, or I think we understand quite a bit, what happens with plants and trees under drought. So we know that the water transport of trees, the hydraulics of trees, are at the center of drought responses of plants. We understand pretty well that when facing drought, plants tend to first try to control water fluxes. They typically do that by closing stomata. As you can see in the figure in the right, stomata is closed as we get to the right, which involves basically drier and drier conditions. There is a point in which it's not only stomata responding, but also there will be cavitation and embolism in the xylem of plants that results in the loss of hydraulic conductivity, so the loss of the capacity to transport water in plant stems. And this eventually can be associated with leaf shedding, for example, which in some cases is part of a strategy to face droughts. In other cases, it's just the response that plants can afford. And eventually, if this situation, if the intensity of the drought is even more intense or it's maintained during a long time period, plants might dry out completely, they might dehydrate and die. And we do understand also the mechanism of this mortality process, I would say, reasonably well. We know that this mortality process involves both the water and the carbon side of things, so water and carbon economy. Even if we know that hydraulics, water is always involved in this, and carbon is not always involved directly in the mortality mechanism, but it's clearly affected by drought. So we know that when plants close stomata, they typically sooner than later also disconnect from the soil. This can be due to some hydraulic disconnection, for example, due to xylem embolism, again in the roots, or it can be due to a physical separation between the roots and the soil, or by a very steep increase in the hydraulic resistance of the soil. But in any case, very frequently plants, as conditions get drier, they disconnect from the soil, then they are left with basically the water pools they have on the plant tissues. Even when stomata are closed, we know that there are residual water losses through leaves, through stomata, which are not completely, I mean, they have some residual conductance, or through the cuticula. And at some point, if the conditions get even drier, there will be hydraulic failure in the xylem of the main stems or branches. And eventually, this will result in individual cells, tissues, meristems dehydrating and the plant dying. So we do understand quite a bit these mechanisms. We know that also, of course, they interact with other stress factors like biotic agents. And we do know that as conditions get drier, the role of pools of water, pools of carbon, the reserves, the storage becomes more and more important because the connections with the environment get dimmer. And one would think that with this, we should be able to actually predict when these mortality events will occur. And actually, there has been some success at this. This is a study by Bill Anderegg and collaborators, which is already almost a decade old, in which they basically compared the species that at the same site, species that had experienced mortality with species that survived drought events using the global mortality dataset. And they showed actually that hydraulic traits like this P50, which characterizes the hydraulic vulnerability of plants, or the hydraulic safety margin, that is simply the distance between this P50 and the water potentials, the internal stress experienced by the plant, both were a significant explanatory factor of this mortality. So plants that actually died, species that actually died, were either more vulnerable to embolism or they showed narrower safety margins, which is consistent with our understanding and quite encouraging. More recently, we used these similar ideas to predict mortality due to drought, or the risk of mortality due to drought at the global scale. In this study, we took basically the distribution of 40,000 Buddhist species, and we either used hydraulic data from hydraulic databases or used these databases together with phylogenetic and environmental information to impute the data for the species for which we didn't have information. And then we combined this into maps of hydraulic traits at the pixel level. So we could really look at the distribution of traits in each pixel at the global scale and then relate these distributions to observed mortality events, again, using the global mortality dataset. And we saw that actually our models were quite predictive, and we were able to use machine learning to really extrapolate these results into global maps of mortality risk due to drought based on these hydraulic traits, which look broadly credible, and they highlight areas that actually have experienced mortality. Most importantly, these models show that the most important explanatory factors, which you can see on the top right, actually relate to hydraulic traits, in particular, the number of species with negative hydraulic safety margins. So the number of species experiencing water potentials close or beyond the P50 was the most important explanatory factor of this mortality risk, which is, I believe, an important result. But at the same time, in all these studies, in all these papers, the percentage of variance in mortality really explained by hydraulics or by these models was relatively low. And we know that even when we incorporate these hydraulic traits and hydraulic processes into process-based models and use that to predict mortality at the regional scale, like in this case for the United States using the FIA dataset, actually these models are not very good. Again, they show significant effects of hydraulics, like in this case, maximum predicted loss of hydraulic conductivity. But the predictive capacity of these models remains very, very low, as you see here, for many of the most abundant species in the US. And this has led to several papers discussing why is it that mortality is so hard to believe, particularly considering that we have a reasonably good understanding, or many people, including myself, think that we have a reasonably good understanding of the mechanisms of mortality. And of course, there are many reasons of why this is the case. And here there is a summary of several of the complications that contribute to this complexity. Of course, when we want to relate processes or traits to performance, mortality, drought responses at the community scale, there are many many steps that we need to, many jumps that we need to make to actually do this connection. First complication is that there are many traits actually involved in mortality, in tree mortality. It's, of course, the hydraulic traits I just mentioned, but of course these traits operate in a context of a suite of traits that each individual plant or species has. And for example, things like rooting depth, for which we typically have very, very, very limited information, or many other parameters related to size, allometry, allocation, etc., have a very strong impact on mortality risk. And this is very difficult to incorporate in meaningful ways in models, sometimes due to understanding of how all this coordinates, sometimes due to just data limitations, as in the case of rooting depth I mentioned. The second thing we really don't understand very well is trait coordination. So all these traits, as I said, cannot be interpreted in isolation. And the way these traits covary and coordinate at the phenotypic level, so at the individual level, is to some extent still a matter of research. Then, of course, these traits and the way these traits are assembled, are coordinated, is not something that is constant, that is set. They vary a lot, even within species. They vary in space because there is local adaptation, there is plasticity, so they can also vary over time, sometimes substantially. And to make things even more complicated, it's not only about the specific value of a suite of traits in a given location in a given time, but it's really more about how these trait values relate to the environmental factors that determine exposure for the plant. For example, to the soil water potentials to which this plant or the rooting system of a given plant is exposed. So I think that considering all this together gives an idea of why really making these predictions is actually complicated, even if the mechanisms are reasonably well understood. And I would say that there are maybe three key areas in which more research is needed really to improve our modeling, our ability to model and predict mortality. One is improving the way we characterize drought exposure. And this is very challenging because the key variable is water potential, soil water potential, and plant water potential. And this is something that until very recently we were able to measure only mostly punctually. So we have very few measurements over time with a with really not a very good temporal density over time of measurements, nor a very good coverage in space. This is actually slowly changing now, first due to some methodological developments that I will mention later in the talk, but also because there is this network, PSINet, that has been recently created, in which we are assembling water potential data worldwide. And this basically involves getting all the data that has already been published, and I'm sure many of you have been involved in this initiative, but it's also about creating a community of researchers that share data and expertise about the measurement of water potential in soils and plants. So I hope that with this, we will be able to move forward in this characterization of exposure. As I said, it's also about trait variation, and I think we are just starting to scratch the surface over the understanding of trait variation within species, either in space or over time. So here in the left, you have an example of a study we made a few years ago already in the northeast of Spain, in Catalonia, in which we sampled 105 different forests and look at how trait values that for the different species in these plots actually varied as a function of a measure of climatic water availability, which in this case is the ratio of precipitation over potential evapotranspiration in the x-axis in all the plots. Each panel corresponds to a different trait. I will not go through all of them, but just to give some examples. So we, for example, observed that for P50, so for this vulnerability to xylem embolism, there is an adjustment in space across species. So the species that live in wetter areas, which are the ones with higher P over PET in the right, actually are more vulnerable than the species from drier areas. This is quite, makes a lot of sense, and it was expected. But we did not see any adjustment within species, which is consistent to several other studies working with hydraulics. On the other hand, when, for example, looking at the same for the turgor loss point, we saw both an adjustment across species, which you see here in the black line, and also an adjustment within species, less steep, which also makes sense, which you see here for each of the studied species in different colors. And there has been recently an effort to synthesize all this data. This is presented in this plot you have to the right of the slide, in which we conducted a global meta-analysis of hydraulic trait plasticity, and you have here some of the most common hydraulic traits: pre-dawn water potential, midday water potential, the turgor loss point, P50, stem-specific hydraulic conductivity, leaf-specific hydraulic conductivity, and the Hoover value, which is the ratio of subboot cross-sectional area to leaf area. And this is the distribution of all the measurements that have been reported or we could find for each of these variables. And you could see that the distribution, the shape of the distribution actually is very different depending on the variable. And as we know, for example, for P50, it's really very narrow, so there is not a lot of variability within species. So the capacity of adjustment of this variable is quite low, whereas, for example, the changes in water potential can be huge. And this is actually why plants are at increasing risk as conditions get drier, because the water potential gets much more negative, but the P50 does not have the capacity to track this change in water potential. And this is quite interesting because we can compare the different traits. We can also see that things like allocation, like the Hoover value or even the conductivities of the xylem, are more plastic than P50. So I believe we are moving forward in understanding these adjustments, but still I think it's an area that really requires more research. And then the third element I would like to highlight here is really our lack of understanding of coordination, really how all these traits coordinate at the level of individual plants to define a given response to drought or to establish the vulnerability of a given species or population to drought. And this is an example of a study led by José Alberto Ramírez Valiente using Pinus pinaster as the model species, and basically comparing how different traits, including hydraulic traits and other size or allocation-related traits, even growth you have here, in two common gardens, one in a temperate area, one in a Mediterranean area. And again, I repeat, this is for the same species, Pinus pinaster, and this is just a typical trait network. So the edges you see simply correspond to the significant relationships, solid lines, positive, punctuated lines, negative relationships. And what is striking to me, and actually is also reflected in several other studies of trait networks, is that really these networks look completely different. So the way traits correlate to each other and coordinate, at least the traits we measured here, and to the extent that we could assess this coordination, is strikingly different between two common gardens that simply they differ only in the climate, but it's the same species, plants of similar size, otherwise similar conditions. So it's really, and I think we are very far from understanding why is it that we here have this network, and when we go to a drier and hotter situation, we get this network that is completely different. So again, I think this is an area that requires quite a bit of research effort. And maybe to summarize this part of the talk, I think that the bottom line here is that we really still don't understand what are the strategies that define drought response strategies, in particular with regards to mortality. So we don't completely understand yet how the difference in exposure and the way plants limit exposure, which have a lot to do with the plants regulate their water status, their water potential, for example, and with concepts like isohydricity, etc., how this is related to different traits that together coordinate and determine these responses in terms of the physiological fluxes, for example, and how they change over time under a drought. Okay, so reaching this point, I think it's important to also question ourselves, to maybe stop for a while and question the type of approaches we've been using. When I say we, I refer to myself, but also to the community working on forest mortality. I think that we've been dominated, and I think there are good reasons for that, by very reductionistic trait-based approaches, in which basically we have the underlying assumption that we can dismantle our system in very small pieces that we call traits and we can measure, and that then we can put together all these individual traits and we will reassemble something that it's not only similar to the system that we initially wanted to study, but that actually will be doing things that are exactly the same to the system we are studying. And of course, I mean, I'm not going to say that trait-based approaches are not useful or good. They are extremely and have proven to be extremely powerful. But I think that quite frequently we use them, or sometimes we use them without realizing what are the implications or the assumptions they are based on, and also without thinking that there might be other ways of approaching the same problem. Now, if we compare these two DAGs, the let's say real DAG on the left and this automatic DAG on the right, we can say that they do the same. I mean, they look the same to some extent. Probably the automatic DAG in the right can do some of the things that the real DAG on the left can do. But it would be quite foolish to think that the DAG on the right is able to predict, or anything in the DAG on the right will be able to predict the behavior of the DAG on the left. For example, the DAG on the left might just fly away 3 seconds after the photograph was taken, whereas the DAG on the right will certainly not fly. And this has a lot to do with the difference between explanation and prediction. I think that trait-based approaches are very good at explaining things, but they are not necessarily that good at predicting things. And this is very common in any complex system in which we don't really have the capacity, at least at present, and maybe we will never have the capacity to describe the system with the level of detail required to make good predictions based on a given description and explanation of the system. And I think we would all agree that predicting mortality is a complex problem. This diagram you see here has nothing to do with mortality. It comes from health sciences, and I think it has to do with the prediction of obesity, but doesn't really matter. I think that if we did a very detailed diagram of all the variables and factors and processes involved in forest mortality, we would end with something that would be at least as complicated and complex as the one you see. And as I said, in these cases, explanation and prediction frequently are not the same. And I think that also connected to this, we need to think about the scale at which things happen and the scale at which we measure things relative to the scale at which we want to make predictions. Because we know very well in ecology that, of course, we have all these levels of organization, and in trait-based approaches, we tend to have the perspective that we can go from the bottom to the up to the top. No? And we take measurements at a small scale, maybe at the tissue or even within the tissue. And we want to connect these more or less directly to responses at the individual level, or maybe sometimes at the ecosystem scale. And this is all good, but we need to be aware of the fact that there are also many causation loops that come from the bottom to the, from the top to the bottom, that can come from the ecosystem to the individual, from the individual to the tissue, etc. And really to understand how these traits again coordinate and determine any response at the individual plant level, we need to understand both these directions of causality, the one that goes from the bottom to the top and the one that comes from the top to the bottom. This can be very complicated because a priori there are many levels of organization that are relevant, but I would argue that if we want to predict mortality, there is one level of organization that is critical because it's at the level at which we want to make the predictions, at which plants either die or survive, and this is the individual level. And I am, of course, not the first one to say that. No, there are several people in the past, like in this paper that you have here, that have claimed that we need, or that a tree-centered approach is going to be particularly helpful if we want to predict how climatic extremes will impact plants. And this also connects to a philosophical approach that is called the middle-out approach, in which you center from the level of organization you believe is more relevant to the question you are asking, and then you basically expand from this level upwards and to the bottom, instead of basically coming from the bottom or coming from the top. And I think that's potentially a very useful view to take in ecology. And this is to say that actually, when we focus at the individual tree level, there are many examples of empirical approaches that don't use traits, or at least not traits in the common sense of the word, and that don't use process-based approaches that really want to explain things, but that can be quite successful at predicting mortality. This comes from a meta-analysis we did many years ago with Maxime Callaret and Lucia de Soto and many other people, in which, for example, and this has also been shown in many other studies, that plants, trees in particular, tend to start growing less as they approach a mortality event. So in other words, if you see a tree that starts growing lower than the trees that are with which it's growing together, it's quite possible that this tree will die, or at least will be more vulnerable to future droughts. And we even showed, for example, that the resilience, the response to individual droughts in the past was a good predictor of future mortality risk. This is what you see here in the right. And that actually the component of resilience that was more relevant to predict future mortality risk was different for angiosperms, in which the main factor that predicted mortality was the initial impact of the drought, relative to gymnosperms, for which it was actually more important the recovery after drought, so that the individual trees that took longer to recover from a given drought were the ones that have higher chances to die in a future drought. And this was always comparing trees, individual trees of the same species growing in the same location. So it was a controlled paired design. And it is also true, and this is work in which I was not involved, this is Frank Sterk and collaborators. It's even true that sometimes these mechanistic approaches are even more explanatory than, these empirical approaches are even more explanatory than mechanistic approaches. No? This is a recent study in which they compared hydraulic traits like the hydraulic safety margin with more empirical approaches based on growth or leaf lifespan legacies to predict mortality in a controlled common garden study. And they showed that hydraulic traits were actually not very good predictors of mortality across species, but actually changes in leaf lifespan or growth resilience, as I presented also before, were actually very good predictors. I think this is very important because this makes us realize that, as I said before, the approach that is best at explaining is not necessarily best at predicting. And I think we need to be aware that these two different modes of understanding are happening and are possible. I'm not saying that it's better to use empirical models, because of course with empirical models you are losing a lot in terms of understanding and explanation of the system. But what I claim, what I will be claiming in the talk, is that I think we can potentially do an effort to unite these two views, these two approaches, to get hopefully the best of these two worlds. And one element that can be useful in that regard is that can be used to give a conceptual underpinning to this more empirical approach. It is this concept that has been in the literature for decades of early warning signals. We know that in many systems it's to be expected that when they approach a catastrophic shift, and we can assume that individual mortality is a catastrophic shift in the state of a system, but when systems approach these situations, they tend to have higher variance, higher autocorrelation, which is called as critical, it's termed critical slowing down. And several people, including ourselves, has looked at whether these early warning signals actually can be observed in radial growth dynamics in tree rings. And this is again from this meta-analysis we conducted a few years ago with Maxime Cagneret. And basically we show for different stress factors leading to mortality that for gymnosperms, actually, for all these stress factors, we see that individuals that die actually have higher variance and higher temporal autocorrelation than individuals that survive before mortality. So we could claim that there is a usable early warning signal in this case, but we don't see this for angiosperms, for example, as you see in the black symbols as lines. And this is quite similar to what has been shown in several studies. So if we use tree rings, sometimes early warning signals seem to work as predictors of mortality, but in other cases they don't work. And as far as I know, we really don't understand why this is the case. Of course, there are differences between angiosperms and gymnosperms in the dynamics of growth and growth recovery after drought, but I don't believe we fully understand why this difference happens. I believe that to really get the best of these two worlds, to get the best of the empirical approach and the process-based and trait-based approach, we need to do two things. One is working at the level of individuals, which, as I said, is, I think, the best way of staying at the level of organization that is most relevant for mortality. And the other thing is using state variables that connect as much as possible with the mechanisms of mortality as we understand them. And really the key state variable, the key for understanding mortality mechanisms is the water status of plants. That can be the water potential or the water content of plants. And in that regard, this is, I think, a major step forward from working at the level of tree rings, because here we are working first at much finer temporal scales, and second, we are directly working with the variable that we believe is central at maintaining the homeostasis, the behavior of the tree in terms of its water status. And as I said, this applies both to water content and water potential. These are two variables that I think are useful as descriptors of the water status of plants in the context of mortality. I would claim that specifically for mortality, relative water content or water content is potentially even more relevant, or at least as relevant as water potential, because actually, as we saw at the beginning, the importance of pools become more and more important as plants basically disconnect from the soil, and water content, basically the pool of water content is directly the net balance between water supply and loss, but also because water content actually connects also with the site of the carbon balance of trees, with the carbon economy, because non-structural carbohydrates are a very important element in osmoregulation, and hence they contribute to water retention and to the dynamics of water content in cells and tissues. And of course, another big advantage of really focusing on water status, water potential, water content is that these two variables are not only key in terms of drought responses, they are also key in terms of predicting vulnerability to host and pathogens, to pests and pathogens. And they are also critical in predicting the vulnerability to wildfires, because of course, if the vegetation, if the biomass is drier, they will be more flammable and hence more prone to wildfires. So I think this really places these variables at the center of the prediction, or at the center of a substantial part of what is needed to predict how forests will be responding to climate change type disturbances. And then there are also more methodological, technological reasons why I think we are in a good position in terms of using time series of water content and water potential to study drought responses of trees, and particularly mortality risk. And this is because there are new technologies being developed or being generalized, for example, to measure water content in tree stems, like TDR type technologies, or of course dendrometry. There is more recent development of microtensiometers or optical dendrometry that allows really estimating water potentials at very, very fine temporal resolution and get this time series of water status for long periods of time and with high temporal resolution. All these methods are being implemented in monitoring networks in forests worldwide. I think potentially the first using these networks, at least for stem dendrometry, was the Swiss TreeNet, but there is also the Czech Dendro network. There are several others. We are also developing one such network in Catalonia, in which we are combining stem dendrometry with stem water content sensors, with subfluxes, and with stem water potential measurements. And I think there is a huge potential in putting together all these spatial explicit and at the same time highly temporal resolved measurements of water status in trees. And this is so also because all these measurements, particularly the water content measurements, connect to things we can more and more easily sense using remote sensing. There are several remote sensing techniques, particularly microwave remote sensing, that are sensitive to the water content of vegetation, of canopies. And this really opens the possibility of measuring this water content at spatial scales and with temporal resolutions that have been impossible until recently. And there is already some evidence that these dynamics of vegetation optical depth, which is this measure of canopy water content from microwave remote sensing, can be connected and can be predictive of mortality patterns. For example, you have here an example using the huge mortality event in California in 2015. Of course, there are many challenges ahead. Many of these challenges, when thinking in terms of remote sensing, are methodological in really connecting this BOD with a measure that is meaningful in terms of the water content of plant canopies. I will not get into the details here. I think it's also important to realize that it's not only technological or methodological challenges we will face, we also face conceptual challenges, because all of a sudden, by being able to measure water content in ecosystems, we are faced with the challenge of connecting our understanding of plant physiology at the tissue level with the understanding of plant physiology at the ecosystem level. And this is really not the same. Of course, it's connected, and we can make the connections, and there are ways to do these connections, and there have been several recent studies doing so. For example, studies thinking in terms of ecosystem pressure volume curves, so the relationship between water content and water potential, but not at the tissue level, like in a standard PV curve that we can do on a leaf, but at the level of whole ecosystems and how they can be even developed and conceptualized. But I think there is quite a lot of work to be done in this regard. And I also think that, connecting back to the early warning signals and mortality, there is a lot of hidden information of potential in this very detailed time series of water status that can inform us about mortality, that can be developed into early warning signals. And this is something that has not been done, as far as I know, for water status, but there are examples of similar analysis, for example, for carbon fluxes in ecosystems, in which sophisticated physical analysis of time series dynamics using phase state representations like you have here are used to really understand the dynamics of these time series and, for example, identify dynamics that tend to be observed, for example, before a catastrophic shift on the system, which might be mortality or leaf loss or a change in the dynamic behavior in the physiology of these trees or forests. And this is now starting to be possible at regional scales, I believe. And as I said, this already has been shown for the carbon cycle. So some papers have identified signatures in the dynamics of the carbon fluxes at the ecosystem level that indicate the potential for destabilization of these fluxes at the ecosystem scale. And of course, doing this will require quite a lot of coordinated effort. So it requires a strong community. And this is why I think that the International Tree Mortality Network and all the effort behind the International Tree Mortality Network is quite important if we really want to increase our ability to predict forest mortality. As I said, I think that we need to really focus probably more at the whole tree level, but of course we need also to connect with the levels below and above. And for this, and I will be almost finishing with this slide, for this I believe it's, as I said already a little bit at the beginning, I believe it's very important to make this connection between the International Tree Mortality Network, that, as I said, I think it needs really to be as vibrant, as active as it has been up to now, or if even more if possible, to connect this with efforts that are being also developed at smaller scales, at regional scales, at the level of countries, in which they are also we are also organizing, or people are also organizing, to really put together all the community working on forest mortality. And I just wanted to introduce, as an example, the Spanish REDEC, in which basically all the community of people working on forest mortality in Spain were put together. It's a rather new network. We are not even 2 years old, but it has really been very, very, very active in the last couple of years. There are several commissions working on many of the same topics in which the International Tree Mortality Network, of course, is also interested, like databases, mortality attribution, monitoring, mechanistic modeling, but also connecting to things that maybe are a bit more difficult from the perspective of a global network like the ITMN, like really linking with management, decision-making, etc., at these regional and local scales, in which actually many of the decisions in terms of forest management and policy are made. So I think it's really important that we make these connections, and we try to also connect to use the ITMN as a global umbrella for all these efforts that are being developed at the moment. And I would like to finish presenting saying a few words about this collaboration with an artist, Paula Bruna, in which she's using the sounds or the ultrasonic emissions of cavitation and embolism in plants to study also the mortality of trees and forests, but from a very different perspective to the one we normally use in our community. So she's connecting these ultrasonic acoustic emissions with flamenco with the music of flamenco. And she's basically using these connections to create a dialogue or to think about the possibility of a dialogue between the trees and people in terms of how trees and forests are responding to climate change by using flamenco as a language, as a common language that can basically approach these two worlds and actually also change maybe the way we connect with the trees and with the mortality of the trees. So I think that maybe also being a bit, opening a little bit our approaches to these other ways of looking at tree mortality can be quite fertile in terms of thinking of ways of understanding the systems we are trying to work with. And with this, I will conclude by also thanking my research group at CREAF and the Autonomous University of Barcelona, and you for your attention, and I will be very happy to answer any questions you may have. Thanks very much.
Yeah, Jordi, perfect. Thank you very much for this impressive, comprehensive overview of the current understanding of the causes of drought-induced tree mortality. We have several questions coming up. I would like to start with one from Steven Jansen, and it also goes kind of in the direction of, oh, I was raising the hand instead of whatever, goes in the direction of filling the water potential gap. If we would have many cheap sensors available to measure traits related to tree or forest resilience to stress, would we really make progress? Will we be measuring the right traits, or would we just overcome some temporal or scaling issues? It's a provocative question, I would say, Stephen.
No, it's a very good question, I think, and it really connects, I think, very much with some of the message part of the message I wanted to give in the talk. So I think the problem we face is not only a data problem. So it's partially a data problem, I think. So of course, more data is helpful in many cases, but I think we face many types of problems. And I actually personally believe, and I think there are more philosophical reasons to believe, that even with the best data we could have, we would still face issues at predicting such a complicated and complex thing as forest mortality. But I think that also the value of data is very different depending on the trait you measure. So a little bit my message here was that I believe that water status in particular is such a key plant response. I wouldn't necessarily call it a trait. I prefer to use traits for more stable things, but well, we can argue about this. But in any case, it's a variable that I believe contains so much information at the right level, because that's, well, or we can say that it connects to the individual level quite easily, and also with the right resolution and with the right connection with our understanding of the mechanism of mortality, that really, I believe it's, if I was to choose, I think I would put my effort, I would invest in measuring more water potentials continuously. That is my feeling. But of course, yeah, I mean, it's a very good question, yes.
But I think we all agree that we need more data on water potentials from the field. But just shortly a question of myself. We are now able to predict the time of death in saplings in experimental settings. So we know what traits to measure to predict that. Why can we not upscale that to the forest side, to the stand side? And I'm really wondering if the quality of data that we have, if that's really sufficient to widen it, to really go to the stand or ecosystem scale. What do you think?
Again, I think it's part of the problem has to do with the data, as you say, but part of the problem, I think, has to do with the huge amount of processes that are involved in the field that we can control to some extent in the greenhouse or in more smaller scale controlled experiments that make the problem much easier to tackle. So I think I'm not sure that, as I said before, I'm not sure it's only a matter of measuring more traits or measuring them better. That would certainly help, but I think we still will face limitations of trait-based approaches, even in best case scenarios in which we can measure all that is conceivable. But yeah, this is arguable. I mean,
so
So that's why I think that measuring something that is already an integrative of the response of the plant, like the dynamics of water content or water potential, it's a much more integrative response. So I think it conveys a lot of information that can, at the very least, complement the information we get from the traits themselves.
A more specific question by Bill Enderek. Why do you think that plasticity of Ks is as high as Kl and the Huber value? I would tend to think of the leaf area allocation related rates to be the most plastic.
So that is a good question, no? Because I tend to think that these three traits, Ks, Kl, and the Huber value, actually are very tightly coordinated in general. So it is true that if one thinks from the perspective of what is easier for the plant to change, well, it seems that changing the Huber value and hence Kl is easier than Ks, which has to do more with anatomical structural traits. But because they are so tightly linked, I think that in the end, they tend to be coordinated and show similar variation. And actually, of course, Ks is also related to growth. And by growing, the plant has a sort of a shorter term response in which it can adjust also to these changes in leaf area, etc. So yeah, I think I don't have a very good answer, but I think it really has to do with the coordination of the three.
Harold Bookman would like to know, would you think that we could perhaps substitute detailed knowledge on tree water status by tracking the state of soil moisture and atmospheric demand, and thus make predictions of mortality risk?
I guess there is a lot I think that we can know from measurements of soil water content and atmospheric conditions. But I would be very hesitant to not consider the measurements on the trees, because I think that the way trees integrate these, so tree water potential dynamics or tree water content dynamics, this really is the integration of soil water content conditions, atmospheric conditions, through the lens of the specific combination of traits that makes this individual tree. And to me, this is the key to understand why a given species, individual, will survive or not a given drought event. No? Because otherwise, if it was all about the environment, the environment, we can make predictions from the environment. I think they will always be very limited, because if it was all about the environment, all the individuals in a given location would do the same, which we know is not the case. So I think there is probably a lot of untapped potential there, as Harold says, but at the same time, maybe because I am an ecophysiologist, I would really be very hesitant not to consider the level of the measurements on the plant itself.
Thanks. Let's continue the discussion.
It's quite weird, by the way, that I read out your questions, even so you are in the audience, but it's a consequence of now that we have another format than usually. Nevertheless, Giorgio Vacciano would like to know, or asks, what is the current status and predictive power of process-based forest simulation models regarding the prediction of plant water status? What are the next steps that need implementation?
My experience is that predicting, process-based, trait-enabled hydraulic models are getting better at predicting water status, but they are actually better at predicting typically fluxes than water status, which is actually quite interesting. And this is possibly, I would say, likely related to the fact that they are not also very good at predicting mortality, because I would argue that water status is really key to the mortality. So I think it's actually a very good question in that regard, because it points to something that is missing or not done very well by mechanistic models at present. What are the next things? So I think that, again, so why is it difficult to predict water status? Because for the same reason why it is informative to measure it, because it integrates over so many different processes that actually happen on plants and that we only understand partially. So in terms of what are the next steps, well, I think that probably the main, if I had to say only one, I would say knowing more about rooting depth and rooting depth distributions. That, I think, is probably the most important limitation of many of these models. And this is a data limitation, actually.
It goes in the direction of another question. Susan Quick is wondering whether we better can use the data we already have than rather extending the data range. Maybe our models need to be better verified for thresholds and tipping points, as you suggest.
Yeah, I think we probably need to do both. No? So I think it's true that we have already a lot of data and we need to make use of this data. And I think there are efforts like I presented, the PsyNet or SubFluxNet or FluxNet or many other communities, networks built around specific types of data and collecting these data globally and promoting the measurements of these variables globally. But at the same time, I do believe that we also need to get more of this data, at least, or as I already said several times today, in terms of water status, which I think is probably the one that is for which the added value of getting more data is higher, because it's very relevant on the one hand and is possibly the one we have less information of. If you compare the amount of data we have on water potentials with the amount of data we have for carbon or water fluxes, it's really very different.
Yeah, I fully agree. Tamir, should I read it out or do you want to go ahead yourself? I'll just continue. So Tamir is asking, regarding the global map of drought-induced tree mortality risk, does it integrate climate predictions or specifically the species sensitivities?
It's a map of the current mortality risk. So it integrates the climate, but the current climate. So the current climate and current soil conditions that determine the local exposure. But we did not make the effort of making future forecasts of this risk. Also because I think we were already quite brave at showing these maps, because I think they are useful, I hope, and meaningful, but at the same time they are very uncertain. So I think it's already pushing it a little bit, showing it for the present, but moving them to the future. I think we need more information to do that in a meaningful way.
So I would say the last question from Bill Anderek. If we could predict plant pre-dawn water potential across space and time, would we be most of the way there in terms of predicting drought-induced mortality?
So you said pre-dawn water potential.
Yeah, this is a good question, because I thought, do we need pre-dawn or midday? But pre-dawn.
I think it would be a huge step forward, but I still think that there are things that happen in the plant that really connect this pre-dawn with midday that would be, it would be very difficult to make good predictions without considering this. Again, yeah, I guess that if I believed otherwise, I would not be working as an ecophysiologist, but of course, yeah, being able to know spatiotemporal variability in pre-dawns would be a major step forward for sure. Yeah.
And with that, I think I would end the question part here. Thank you very much again, Jordi. It was really a pleasure that you relaunched our seminar. The next speaker will be Ruiling Liu on April 14. She's from Belinda Maitland's group, and she will talk about rising tree mortality across Australian forests. And then I hope to see many of you in 3 weeks again.